I hope the slides are visible.
Sir. Sir.
Okay. Thanks for the confirmation. Okay, we will start with the today's topic. Next quality guideline we will see, that is a Q8 guideline. It is about pharmaceutical development. Okay, so pharmaceutical development is, I think, most of you aware that whenever any project is there in the industry, any new formulation needs to be developed, then which steps to be followed while performing that formulation development. That steps are given, and how to record or how to do the documentation. How to do the documentation of that activities, that is given in the Q8 guideline. मतलब फॉर्मुलेशन जो हम डेवलप करने वाले हैं उसका वो कैसे करना चाहिए और वो कैसे डॉक्यूमेंट के जरिए अथॉरिटी को सबमिट करना है, वो डॉक्यूमेंट का स्ट्रक्चर Q8 गाइडलाइन में दिया है. So here is a objective. This guideline describes the suggested contents for the 32P2 section of a regulatory submission in the ICH-M4 Common Technical Document format. जो CTD डोजियर हम कहते हैं कि वो CTD डोजियर, तो उसमें एक सेक्शन रहता है 32P2. वो सेक्शन में हमको फार्मास्यूटिकल डेवलपमेंट रिपोर्ट अटैच करना रहता है. So this pharmaceutical development report is generated at the F&D department. F&D means Formulation Development Department, also called as a Research and Development Department. So these teams generate this document, and the signed version of this document is attached directly in this 32P2 section of the dossier. The pharmaceutical development section provides an opportunity to present the knowledge gained through the application of scientific approaches and quality risk management to the development of a product and its manufacturing process. So whatever the knowledge we gain through the development process, that knowledge is represented via this PDR document. It is first produced for the original marketing application and can be updated to support new knowledge gained over the life cycle of a product. So at the first time, मतलब जब ये product initially develop होता रहता है तभी ये first time ये document generate होता है. और जैसे डॉक्यूमेंट मतलब जैसे प्रोडक्ट में कुछ चेंजेस होते हैं कि बाबा सपोज इनिशियली जब ये डॉक्यूमेंट जनरेट हुआ तभी प्रोडक्ट का डिस्क्रिप्शन राउंड शेप था, लेकिन बाद में ड्यूरिंग द क्वेरी रिस्पॉन्स या ड्यूरिंग द वेरिएशंस आफ्टर अप्रूवल ऑफ द प्रोडक्ट there is some changes in the appearance of the product. Then accordingly the pharmaceutical development report also needs to be updated. So in this way it keeps on updating if you do the changes in the product itself. Next is the aim of pharmaceutical development is to design a quality product and its manufacturing process to consistently deliver the intended performance of the product. Okay. The information and knowledge gained from the pharmaceutical development studies and manufacturing experience provide scientific understanding to support the establishment of the design space, specifications, and manufacturing controls. So with the help of the pharmaceutical development report, we can build a quality product, and also we can build a manufacturing process which help us to consistently deliver a quality product. तो ये दो ऑब्जेक्टिव्स है फार्मास्यूटिकल डेवलपमेंट की. एक तो क्वालिटी प्रोडक्ट देना और उसके बाद एक मैन्युफैक्चरिंग प्रोसेस जनरेट करना या प्रिपेयर करना जिससे कि कंसिस्टेंट लेवल पे, कंसिस्टेंट बेसिस पे क्वालिटी प्रोडक्ट मैन्युफैक्चर कर सकते. सो दैट इज द एम. Next, what is the design space concept? The multidimensional combination and interaction of input variables, example material attributes and process parameters, that have been demonstrated to provide assurance of quality. So it is a theoretical concept. Design space means it is a combination and interaction of input variables. Input variables means what? API है, excipients है, उसके बाद जो हम इक्विपमेंट्स यूज करने वाले वो इनपुट वेरिएबल्स हो गए, बराबर. उसके बाद प्रोसेस पैरामीटर्स. प्रोसेस पैरामीटर्स मतलब जो हम हार्डनेस है, फ्राइबिलिटी है या थिकनेस है. उसके बाद ये जो प्रोसेस पैरामीटर्स है, the combination of these input variables and process parameters is considered as a design space, is combinedly considered as a design space. Working within the design space is not considered as a change. Suppose you have developed or you have established already a design space. Already a design space means you have established some input variables and also the manufacturing process or process parameters. If the company is working within that established parameters, then it is considered as no change. But after the approval, if the company is deviating from that design space, means they are changing the excipients, they are changing the process parameters, then it is called as a change, and that change should be get first approved from the authority via a variations process, and then only it can be implemented in the actual industry, pharma, actual manufacturing or pharma industry. So that is called as a change, and which is routed through the variation process. Design space is proposed by the applicant and is subject to regulatory assessment and approval. So during the initial process, during the initial phase, when the pharmaceutical development report is generated, that report first needs to be approved by the authority. That's why we submit it in the dossier. जैसे ही वो अथॉरिटी अप्रूव करती है, उसके बाद हम फिर वो इम्प्लीमेंट कर सकते हैं कमर्शियल बैचेस में. और उसमें भी अगर कुछ चेंज करना है तो हमको वेरिएशन करना है, और वो वेरिएशन अप्रूव होने के बाद फिर हम वो चेंज इम्प्लीमेंट कर सकते हैं. So this is called as a design space. Any doubt till now? Have you understood the pharmaceutical development overall idea? Means we will go in deep in the next slides, but in general idea.
Yes, sir.
Okay. Yes. So here, pharmaceutical development should include at a minimum the following elements. So what are the contents of the pharmaceutical development report that we will see? Defining the quality target product profile as it relates to quality, safety, and efficacy, considering the route of administration, dosage form, bioavailability, strength, and stability. Abhi, what is meant by QTPP? Why it is required? Can anyone explain here? What do you understand from QTPP? Yes.
QTPP is required because it means to ensure a product meet the quality standards or not. Process of pharmaceutical development.
Quality standards. Okay. Uh, yes. Yes, you are right. Quality standards. Whenever we need to start any pharmaceutical development, we need to set some standards that whatever formulation we will develop, that should match with the standard. Okay. So first, to set that standards, we need to set the quality target product profile. Because the QTPP is related to the quality, safety, and efficacy, considering the route of administration, dosage form, bioavailability, strength, and stability. So, relating to all these parameters, the QTPP should be established. मतलब कौन से रूट ऑफ एडमिनिस्ट्रेशन का हमको फॉर्मुलेशन डेवलप करना है, उसका डोसेज फॉर्म क्या होना चाहिए, what should be the bioavailability, strength, and stability, what should be the shelf life of that product. So all these standards first should be set कि हमको क्या अचीव करना है. वो अगर स्टैंडर्ड्स अवेलेबल होगा तो ही हम वो मतलब एक सेट स्टैंडर्ड होगा तो हम वो अचीव करने के लिए ट्राई कर सकते हैं. अगर आपके पास कुछ सेट स्टैंडर्ड ही नहीं हो तो आप अचीव क्या करोगे. ऐसा है. इसके लिए QTPP is important while initiating any formulation development. Okay. Identifying potential critical quality attributes, CQs of the product, so that those product characteristics having an impact on product quality can be studied and controlled. So here, potential critical quality attributes we need to identify of the product. अभी product की critical quality attributes क्या हो सकते हैं? Related to the shelf life also. अगर suppose shelf life या प्रोडक्ट अगर ज्यादा सोल्युबिलिटी हमको इंक्रीज करनी है तो सोल्युबिलिटी विल बी अ क्रिटिकल क्वालिटी एट्रीब्यूट फॉर द डिसोल्यूशन. लाइकवाइज मतलब माइक्रोबियल ग्रोथ, अगर माइक्रोबियल ग्रोथ के लिए प्रमोट कर रहा है ड्रग प्रोडक्ट तो उसको कैसे कम किया जा सकता है, दैट विल बी अ क्रिटिकल क्वालिटी एट्रीब्यूट. Critical quality attribute varies based on the dosage form we are developing. Suppose एक chewable dosage form अगर हम develop कर रहे हैं तो hardness is the critical quality attribute in that case, because hardness should be more for the chewable tablets. They should not be easily break into the smaller parts, because it should be chewed, and that's why the hardness of the chewable tablets is a critical quality attribute. For the low dosage form, low dose products, that is a digoxin we can say, or sodium nitrite, those are very less in dose. So in that case, a critical quality attribute is uniform distribution we can say, or यूनिफॉर्म डोज डिलीवर अगर करना है तो फिर प्रॉपर मिक्सिंग होना जरूरी है. सो दैट वी नीड टू आइडेंटिफाई. देन determining the critical quality attributes of the drug substance excipients and selecting the type and amount of excipients to deliver a drug product of the desired quality. So here we need to identify the CQA of the drug substance and excipients. Here CQA of drug substance may be the particle size we can say. अगर पार्टिकल साइज कुछ प्रोडक्ट अगर जो सपोज बहुत कम सोल्यूबल है, these are very less soluble, then we need to focus on the particle size. And polymorphism Suppose a drug substance exists in 2 or 3 polymorphs, then the polymorph which is having the desired therapeutic effect, that we need to observe. So that is the critical quality attribute. Then excipient में भी कुछ excipients का particle size important होता है for the final product performance. So in that case also particle size plays a very important role and it serves as a critical quality attribute. So in this way, we have to identify the CQA of the drug substance and excipient also. The next is selecting an appropriate manufacturing process. So this is very much important, because if you fails to develop a proper manufacturing process, even if the composition is very much suitable, the final product will not be a quality product. So that's why the manufacturing process should be appropriate. Then defining a control strategy. Control strategy means during the manufacturing process, what are the control steps? What are the in-process tests we are doing to check whether the product is manufactured as per the required standards or not? That is called as a कंट्रोल स्ट्रेटेजी. मतलब सपोज अभी हम एक सॉल्यूशन डोसेज फॉर्म मैन्युफैक्चर कर रहे हैं, तो बीच में हम मिक्सिंग प्रॉपर हो रहा है कि नहीं, या पीएच बराबर मेंटेन हो रहा है कि नहीं, ये हम चेक कर रहे हैं कि नहीं. दैट इज कॉल्ड एज अ कंट्रोल स्ट्रेटेजी. स्टराइल प्रोडक्ट है कुछ, तो हम जो स्टरलिटी प्रॉपर मेंटेन हो रहा है कि नहीं ड्यूरिंग द मैन्युफैक्चरिंग प्रोसेस, देन माइक्रोबियल ग्रोथ को कम मतलब दैट इज eliminated or not, and proper sterilization हो रहा है कि नहीं. That are the control strategy which is applied during the manufacturing process. That also need to be taken into care while performing the pharmaceutical development. So these are the elements which should be considered. First is we need to set a QTPP. Second, we need to identify the CQA of the drug product. Then we need to identify the CQA of drug substance and excipients. Then next, we need to develop a manufacturing process. Then we need to develop a control strategy. So in this way, these are the elements. Any doubt in this slide?
No, sir.
Okay. Next is contents of pharmaceutical development. So now actual जो डॉक्यूमेंट होता है, मतलब फार्मास्यूटिकल डेवलपमेंट रिपोर्ट, उसका क्या क्या कंटेंट्स रहते हैं और वो हर एक पॉइंट के अंदर क्या इंफॉर्मेशन इंक्लूड होनी चाहिए, वो सब इसमें दिया हुआ है. सो वी विल सी वन बाय वन. फर्स्ट इज द 2.1 components of the drug product. Okay, so in this drug substance and excipient, these are the two components. Okay, so first is the drug substance. The physicochemical and biological properties of the drug substance that can influence the performance of the drug product and its manufacturing ability, or were specifically designed into the drug substance, should be identified and discussed. So the physicochemical and biological properties of the drug substance which can influence the performance of the final drug product and its manufacturing ability, that should be discussed. मतलब उसमें होता है कि सोल्युबिलिटी है, उसके बाद पार्टिकल साइज है, फ्लो प्रॉपर्टीज कैसी है ड्रग सब्सटेंस की, इट इज सो दैट. देन पॉलीमॉर्फिज्म है, टैप डेंसिटी, बल्क डेंसिटी है. So these properties which can influence the drug product performance or manufacturing process, those should be identified and discussed. मतलब सपोज अगर टैप डेंसिटी, बल्क डेंसिटी या कॉम्प्रेसिबिलिटी इंडेक्स, ये जो पैरामीटर्स है, ये पैरामीटर्स अगर they are within a good range, then the flow property will be good. Okay, And if it is not good, then the flow properties we need to enhance with the help of excipients. So we need to identify these parameters which can influence the drug product performance, and then we need to discuss the positive and negative points about that. So if positive, then it is okay, but if it is negative, then how we will overcome that during the manufacturing process, that also we need to discuss here. Examples of physicochemical and biological properties that might need to be examined include solubility, water content, particle size, crystal properties, biological activity, and permeability. So these are some of the examples of the physicochemical and biological properties. The next is excipients. The excipients chosen their concentration and the characteristics that can influence the drug product performance. Example, stability and bioavailability or manufacturing ability should be discussed relative to the respective function of each excipient. So here we can say so excipients मतलब कौन से एक्सिपिएंट है, एक तो डाइल्यूएंट है, उसके बाद बाइंडर है, डिसइंटीग्रेंट है, लुब्रिकेंट है, film coating agent hai. So all these ingredients which are to be used during the manufacturing process, the properties of each ingredient, the properties of each excipient should be discussed. And also the function, means why we are using that excipient. This should include all substances used in the manufacture of the drug product, whether they appear in the finished product or not. Compatibility of excipients with other excipients where relevant. For example, combination of preservatives in a dual preservative system should be established. So here we can say whenever we will start any formulation development, we need to first do the compatibility study of API with the excipient. Compatibility study means what? We need to check whether the API is getting properly adjusted with the excipient or not. Properly adjusted मतलब क्या है कि actual में what is the experiment done here? API और excipient एक certain proportion में वो मिक्स किए जाते हैं और वो पेट्री डिश में रखे जाते हैं और that are charged in the stability or placed in the stability chamber at 4075 for 4 weeks. Okay, so after the 4 weeks they are withdrawn from the stability chamber, that petri dish, and it is checked for any changes in the appearance of the API, and it is checked whether the assay मतलब API का कुछ क्वांटिटी कम हुआ है क्या ड्यू टू डिग्रेडेशन और ड्यू टू इंटरेक्शन विद द एक्सिपिएंट, एंड आल्सो द impurities which are there are increased or as it is. So what is the impact on the assay value and what is the impact on the impurities content, that is checked. If that impact is more, then the conclusion is the API and excipients are not compatible with each other in that concentration. Then the F&D person will change the concentration and try, and if the same results again obtained, then the excipient will be changed and other inert excipient will be used. Other inert excipient or other excipients in different concentrations will be used. So in this way, the compatibility study should be performed before finalizing the excipients. So this is very important step before going ahead for the formulation development. Any doubt in this slide? Okay. Next is drug product formulation development. A summary should be provided describing the development of the formulation, including identification of those attributes that are critical to the quality of the drug product, taking into consideration intended usage and route of administration. So here again we need to discuss about the how we means what are the critical quality attributes of the drug product. That needs to be discussed here. The summary should highlight the evolution of the formulation design from initial concept up to the final design. So from the starting to the end, how the formulation will be developed, that should be described here. The summary should also take into consideration the choice of drug product components, example, the properties of the drug substance, excipients, container closure system, any relevant dosing device, the manufacturing process, and if appropriate, knowledge gained from the development of similar drug products. So here we need to summarize the development process which we will follow for that drug product. Okay, a summary of formulation used in clinical safety and efficacy and in any relevant bioavailability or bioequivalence studies should be provided. So here we need to provide the summary of that formulations which will be used in the bioavailability or bioequivalence studies. Any changes between the proposed commercial formulation and those formulations used in pivotal clinical batches and primary stability batches should be clearly described and the rationale for the changes provided. So here, whatever batches we take during the pharmaceutical development, that batches should be the bioavailability or bioequivalence study of that batches should also be conducted on a स्मॉलर स्केल. मतलब पायलट बी स्टडी जो हम बोलते हैं कि कम से कम पेशेंट लेके सिर्फ चेक करना है कि बाबा द फॉर्मुलेशन विच वी आर ट्राइंग टू डेवलप दैट इज डेवलपिंग इन अ प्रॉपर मैनर और नॉट. सो दैट्स व्हाई R&D बैचेस जो रहती है, अभी इसको हम R&D बैचेस बोलेंगे या पायलट बैचेस भी बोलते हैं, तो उसका भी पायलट बी स्टडी परफॉर्म किया जाता है, सो दैट टू चेक or to confirm whether the process is followed is giving us the proper product or not. And if any changes we need to do, then that changes should also be documented in this section. There are two types of studies. One is a pilot B study and second is a pivotal B study. Pilot मतलब जो small scale पे होती है, जिसमें कम सब्जेक्ट्स एनरोल किए जाते हैं जस्ट टू चेक द प्रॉपर प्रोडक्ट इज डेवलप्ड और नॉट. और ये स्टडी किधर भी सबमिट नहीं होती डोजियर में. दूसरी जो स्टडी रहती है वो पिवोटल बी स्टडी रहती है. पिवोटल मतलब वो लार्जर स्केल पे होती है, मतलब एक्चुअल जो बी स्टडी रहती है वो, और उस वो बी स्टडी हम डोजियर में सबमिट करते हैं. और वो कमर्शियल बैचेस जो प्रोसेस वैलिडेशन की जो बैचेस होती है, पिवोटल बैचेस, उस पे वो बी स्टडी परफॉर्म की जाती है और वो बी स्टडी हम रिपोर्ट उसका डोजियर में सबमिट करते हैं. सो इन दिस वे पायलट बी स्टडी एंड पिवोटल बी स्टडी. Then information from comparative in vitro studies, example dissolution, or comparative in vivo studies, example bioequivalence, that links clinical formulations to the proposed commercial formulation should be summarized and a cross-reference to the studies should be provided. Where attempts have been made to establish an in vivo in vitro correlation, the results of those studies and a cross-reference to the studies should be provided in this section. A successful correlation can assist in the selection of appropriate dissolution acceptance criteria and can potentially reduce the need for further bioequivalence studies following changes to the product or its manufacturing process. So here, formulation performance, the performance of the formulation, the performance of the product is also checked with the help of comparative dissolution studies. एक तो रहता है कि बी स्टडी से भी चेक किया जाता है, जो हमने लास्ट स्लाइड में देखा, और दूसरा परफॉर्मेंस चेक करने का वे है कि वो कंपैरेटिव डिसोल्यूशन स्टडी। अभी इसमें क्या होता है कि रेफरेंस प्रोडक्ट जो रहता है, जिससे हम कंपैरिजन करके प्रोडक्ट बना रहे हैं, हमारा जेनरिक प्रोडक्ट, उसका डिसोल्यूशन एक इसमें लगाते हैं apparatus में, और जो टेस्ट प्रोडक्ट रहता है उसका भी डिसोल्यूशन दूसरे apparatus में लगाते हैं। तो कंपेयर करते हैं कि बाबा डिसोल्यूशन जो है दोनों का कंपेरेबल है कि नहीं। मतलब रेफरेंस प्रोडक्ट जो जितने समय में डिसॉल्व हो रहा है उतने समय में ही टेस्ट प्रोडक्ट डिसॉल्व हो रहा है कि नहीं। अगर वो कंपेरेबल है तो वी आर ऑन द राइट ट्रैक, वी आर डेवलपिंग अ क्वालिटी प्रोडक्ट which is in equivalent quality to the reference product. That is called as a compare in vitro in vivo correlation. मतलब ये कोरिलेशन रहता है। मतलब सपोज इन विट्रो जो स्टडीज हम करते हैं, मतलब इन विट्रो स्टडी मतलब क्या, डिसोल्यूशन स्टडी, और उससे हम इन विवो परफॉर्मेंस प्रेडिक्ट कर सकते हैं। इन विवो परफॉर्मेंस मतलब बॉडी के अंदर, बॉडी में मतलब पेशेंट अगर एडमिनिस्टर करता है प्रोडक्ट, तो उस बॉडी के अंदर भी कैसे वो कितने समय में डिसॉल्व हो पाएगा। तो इन विट्रो एक्सपेरिमेंट के जरिए हम इन विवो परफॉर्मेंस कोरिलेट करते हैं। That is called as a in vitro in vivo correlation, IV-IVC. This is a very important terminology, IV-IVC, in vitro in vivo correlation. Based on the in vitro dissolution experience, we establish the in vivo performance of the product. So this is the IV-IVC. So whenever we establish any IV-IVC, that is a in vitro in vivo correlation, then after the approval of the product also, if we do any changes, then we do not need to perform the bioequivalence study. Only the comparative dissolution studies are equivalent for establishing the equivalency between the updated product and the old product. So here is written, a successful correlation can assist in the selection of appropriate dissolution acceptance criteria and can potentially reduce the need for further bioequivalence studies following changes to the product or its manufacturing process. Any special design features of the drug product should be identified and rationally provided for their use. मतलब सपोज कुछ भी अगर एक्स्ट्रा अगर डिजाइन हम कुछ उसमें टैबलेट के अंदर डाल रहे हैं, डिजाइन मतलब क्या कि स्कोर लाइन है, मतलब टैबलेट स्कोर लाइन रहती है बीच में टैबलेट की, तो वो स्कोर लाइन कभी कभी क्या होता है अपीयरेंस के लिए भी होती है, और कभी वो टैबलेट को 2 इक्वल डोसेस में डिवाइड करने के लिए भी होती है। सो इन दिस वे द रीजन बिहाइंड कीपिंग द स्कोर लाइन दैट शुड बी given here if there is a score line. Otherwise, if plain tablet is there, then no need to provide any justification. Next is overages. Overages means in general use of an overage of a drug substance to compensate for degradation during the manufacture or a product's shelf life or to extend shelf life is discouraged. Overage means what? Overage means, can anyone tell what is meant by overage?
Suppose we need to add 100% means we'll add 10% extra, means that 10% will be the average for the active.
Yes. Yes. Right. So whatever extra API we will be used, that extra API is considered as an overage. अभी वो ओवरेज का रीजन कुछ भी हो सकता है टू कॉम्पेंसेट द लॉस ड्यूरिंग द मैन्युफैक्चरिंग, मतलब मैन्युफैक्चरिंग प्रोसेस के दौरान आपका प्रोसेस लॉस इतना है, तो वो प्रोसेस लॉस ओवरकम करने के लिए हम 10% ओवरेज यूज करना चाहते हैं. सो दैट ओवरेजेस क्वांटिटी शुड बी फर्स्ट अप्रूवल, मतलब पहले लेना पड़ता है अथॉरिटी से, बाद में ही हम वो इस्तेमाल कर सकते हैं. क्योंकि जितना आप ओवरेजेस ज्यादा यूज करोगे उतना एपीआई उसमें ज्यादा जाने के चांसेस है और इम्प्यूरिटी ज्यादा जनरेट होने के चांसेस है. तो इसके लिए अथॉरिटी मतलब ये रिस्ट्रिक्ट करती है कि आप कम से कम ओवरेजेस यूज करो. Any overage in the manufacture of the drug product, whether they appear in the final formulated product or not, should be justified considering the safety and efficacy of the product. So कौन से भी ओवरेजेस रहने दो, मतलब वो प्रोडक्ट में, फाइनल प्रोडक्ट में अपियर हो रहे हैं तो भी हमको कंट्रोल करना है, और अगर अपियर नहीं हो रहे हैं तो भी हमको कंट्रोल करना जरूरी है. Information should be provided on the amount of overage, reason for overage, example to compensate for the expected and documented manufacturing losses, and justification for the amount of overage. The overage should be included in the amount of drug substance listed in the batch formula. So whatever overages we are using, that content should be listed in the batch formula. So this is about the overages. For the most vitamin types of जो vitamin type के जो preparations होते हैं, formulations होते हैं, उसमें बहुत ज़्यादा overages use करते हैं, क्योंकि vitamins are likely to degrade early during the shelf life period. So that's why it is the means authority could say, मतलब it is granted that for the vitamin preparations you can use the overages. But for other preparations, we need to provide solid justification for the inclusion of overages. Then physicochemical and biological properties. The physicochemical and biological properties relevant to the safety, performance, or manufacturing ability of the drug product should be identified and discussed. This includes the physiological implications of drug substance and formulation attributes. Studies could include, for example, the development of a test for respirable fraction of an inhaled product. Similarly, information supporting the selection of dissolution versus disintegration testing or other means to assure drug release and the development and suitability of the chosen test could be provided in this section. So here we need to discuss about the physicochemical and biological properties of the drug product. Next is manufacturing process development. The selection, the control, and any improvement of the manufacturing process should be explained. So suppose any means for the R&D batches you have performed a manufacturing, means gone through a manufacturing process, but you have done some changes for the commercial process. So that changes which you have means identified while performing the formulation development study. So that changes in the manufacturing process which are proposed for the commercial batches that should be discussed in this section. It is important to consider the critical formulation attributes together with available manufacturing process options in order to address the selection of the manufacturing process and confirm the appropriateness of the components. Appropriateness of the equipment used for the intended products should be discussed. So here whatever the equipments we have used, मतलब जो हमने मिक्सर यूज किया है, जो ग्रैन्युलेटर यूज किया है, जो ब्लेंडर यूज किया है, जो फ्लूइडाइज्ड बेड ड्रायर रहेगा वो यूज किया है, या फिल्म कोटिंग के लिए कौन सा पैन यूज किया है. So all that equipments should be described here. The knowledge gained from process development studies can be used as appropriate to justify the drug product specifications. So during the process development, whatever the knowledge we have obtained, that should be discussed in this, so that based on that knowledge we can justify that why we have used this equipment for this process, why we have followed this type of manufacturing process. So all that knowledge will be utilized for the justification purpose. The manufacturing process development program or process improvement program should identify any critical process parameters that should be monitored or controlled to ensure that product is of desired quality. So here we have to identify the critical process parameters. So critical process parameters means मिक्सिंग स्पीड है, मिक्सिंग इंटरवल है, उसके बाद एम्परेज रीडिंग है ड्यूरिंग द फॉर्मेशन ऑफ द डफ मास दैट इज फॉर द ग्रैन्युलेशन. देन ब्लेंडिंग स्पीड है, ब्लेंडर का टाइम, ब्लेंडिंग का टाइम है, हार्डनेस, मशीन का हार्डनेस कैसे सेट किया है वो, दैट आर द क्रिटिकल प्रोसेस पैरामीटर्स. Next is the container closure system. The choice and rationale for selection of the container closure system for the commercial product should be discussed. So here mainly what is done in the container closure system, it is directly taken, मतलब जो generic products होते हैं उनका container closure system ऐसे कुछ अलग से डिजाइन नहीं किया जाता. जैसे रेफरेंस प्रोडक्ट ने यूज किया है, मतलब उसने एलुएल ब्लिस्टर अगर यूज किया है तो जेनरिक प्रोडक्ट मैन्युफैक्चरर भी एलुएल ब्लिस्टर यूज करता है, क्योंकि उसको एडिशनल कुछ टेस्टिंग करने की जरूरत नहीं है, और मतलब वो डायरेक्टली वो एलुएल ब्लिस्टर ही यूज कर सकता है. अगर अलग से कुछ यूज करेगा तो फिर उसको फिर से उसमें स्टडीज परफॉर्म करनी पड़ेंगी कि हाँ भाई ये पैकिंग मटेरियल मतलब स्टेबल है कि नहीं, और इसमें प्रोडक्ट की स्टेबिलिटी इंटैक्ट रहती है क्या, मतलब और कितने दिन तक रहती है. सो वो सब स्टडीज परफॉर्म करनी पड़ेगी. इसके लिए जेनरिक मैन्युफैक्चरर्स मोस्टली जो रेफरेंस प्रोडक्ट ने जो दिया है कंटेनर क्लोजर सिस्टम वही यूज करते हैं. Consideration should be given to the intended use of the drug product and the suitability of the container closure system for storage and transportation, including the storage and shipping container for bulk drug product where appropriate. So here the consideration should be given to these parameters while deciding the container closure system. Any doubt till this point?
No, sir.
Okay. The choice of primary packaging materials should consider the choice of materials, protection from moisture and light, compatibility of the materials of construction with the dosage form, including sorption to container and leaching and safety of materials of construction. Sorption means some containers, some materials have the ability to extract the ingredients from the drug product. That is called as a sorption. और लीचिंग मतलब ऐसे कुछ कंटेनर्स रहते हैं जिसके मटेरियल में वो इनहेरेंट प्रॉपर्टी रहती है कि वो खुद का जो मटेरियल रहता है वो लीच करते हैं, मतलब वो ड्रग प्रोडक्ट में उसका मटेरियल का कुछ ट्रेसेस आते हैं. दैट ट्रेसेस आर come into the drug product. So that is called as a leaching. So these two, मतलब ये जो दो ये है, मतलब जो सॉर्प्शन और लीचिंग वो दोनों भी नहीं होना चाहिए अगर आइडियल कंटेनर क्लोजर सिस्टम अगर आप सर्च कर रहे हो तो. And safety of materials of construction. So this also should be taken into care. Example, dropper pipette, pen injection device, dry powder inhaler. It is important to demonstrate that a reproducible, accurate dose of product is delivered under testing conditions, which as far as possible simulate the use of the product. So here, if you are using any medical device for the administration of the product, then it should be checked that it is delivering a means accurate dose or not on a consistent basis. मतलब अगर एरोजोल का प्रोडक्ट है तो एरोजोल जो स्प्रे करने के लिए वो मीटर डोज रहता है, इनहेलर का वो जो पंप रहता है वो हर एक एक्चुएशन पे एक्यूरेट अमाउंट ऑफ डोज डिलीवर कर रहा है कि नहीं वो चेक करना जरूरी है. So that is also a property of the ideal container closure system that should be passed. Then next is 2.5 microbiological attributes. The microbiological attribute of the drug product should be discussed in this section. Microbiological properties. The rationale for performing or not performing microbial limits testing for non-sterile drug products. So here this should be discussed. The selection and effectiveness of preservative systems in products containing antimicrobial preservative or the antimicrobial effectiveness of products that are inherently antimicrobial. For sterile products, the integrity of the container closure system as it relates to preventing microbial contamination. So in this way, we need to go through the microbiological testing of that product. The next is 2.6 compatibility. The compatibility of the drug product with reconstitution diluents should be addressed to provide appropriate and supportive information for the labeling. Compatibility means suppose the company is marketing or developing a powder for injection product. पाउडर फॉर इंजेक्शन प्रोडक्ट के लिए वो डिजोल्व करने के लिए स्टेराइल वाटर ऑफ इंजेक्शन की जरूरत है, बराबर. तो जो भी ये पाउडर्स होते हैं जो डिजोल्व करके हमको एडमिनिस्टर कराने हैं, so the sterile water of injection is given along with them. Okay, so whenever we need to administer that product, we need to dissolve that product into the sterile water for injection, and then we need to check whether it is compatible or not. Means whether the description of that material is changing, whether the there are any increases in the impurities content or decrease in the assay value of the API. So that should be discussed under this compatibility section, and this compatibility section is not applicable for the solid dosage form products. सॉलिड डोसेज फॉर्म के लिए एप्लीकेबल है, मतलब टैबलेट और कैप्सूल के लिए. ये सिर्फ पाउडर फॉर ओरल यूज, पाउडर फॉर इंजेक्शन के लिए एप्लीकेबल है, जिसमें हमको ये पाउडर एक्सटर्नल वाटर फॉर इंजेक्शन में डिसॉल्व करनी पड़ती है और ये फिर एडमिनिस्टर करनी पड़ती है. So in this way, these are the contents of the pharmaceutical development report. I will briefly explain how the QTPP will be established before initiating the pharmaceutical development. QTPP means quality target product profile. So for this, any generic manufacturer company will purchase the reference product from that particular market where the generic company has to market its own product. Suppose it is USA. So the company will purchase the reference product from the USA. Then the company will do the analysis of that reference product and will come to a results, and that results will be the QTPP of that generic product. So this is how the QTPP is said, that is a quality target product profile. Any doubt till now regarding the QA guideline? Okay. Next, these are some questions. So a definition of pharmaceutical development report. What is the definition? Akanksha, are you able to answer this? Yes. Yes.
That is PDR, pharmaceutical development report, means it provides overview of developing pharmaceutical product according to ICH guidelines.
Yes, right. Then what are the components of the drug product? Neha? Yes,
hello?
Gitanjali. Yes.
Excipient and the main drug ingredients.
Yes, API and excipients, right. Then what is meant by comparative dissolution study?
Hello, sir?
Yes.
It means in vivo, in vitro correlation studies.
Yes, right. In vitro in vivo correlation studies.
Where the bioequivalence and dissolution studies will be conducted between the innovative product and the formulation product.
No, in this comparative dissolution study, only dissolution study will be performed. Only dissolution.
Okay.
But in the means bioequivalence study is a different part. Comparative dissolution is a different. In the comparative dissolution, only dissolution profile is compared between the reference and test product. Then enlist the points of a PDR. So this question you will be asked in the interview also, means do you know about the PDR? What is meant by PDR? And tell me the contents of the PDR. This is very much important.
Component of drug product, overages, compatibility.
Microbiological attributes, manufacturing process development, formulation development. So all these are the attributes or the points we can say of the PDR. The next is when the overages are used. What is the reason?
Compensate loss. During the manufacturing process, we lost the API.
Yes, right. To compensate the manufacturing loss, the extra quantity of API is used. That is called as a overages. Right, yes, good. So next is here QTPP is explained. The quality target product profile forms the basis of design for the development of the product. Considerations for the quality target product profile could include intended use in clinical setting, then route of administration, dosage form, then delivery system, dosage strength, container closure system. All these parameters will together constitute as a quality target product profile. Means ये target हमको achieve करना है while we will develop the own product. So रेफरेंस प्रोडक्ट के जरिए रेफरेंस प्रोडक्ट का एनालिसिस करने के बाद उसके रिजल्ट्स जो आते हैं दैट रिजल्ट्स विल बी द क्वालिटी टारगेट प्रोडक्ट प्रोफाइल. सो इंटेंडेड यूज क्या होना चाहिए, मतलब थेरेपेटिक इंडिकेशन क्या होना चाहिए, कौन सी डिसीज कंडीशन के लिए यूज करना है, रूट ऑफ एडमिनिस्ट्रेशन ओरल रहेगा पैरेंटल रहेगा, वो डिसाइड वो एक टारगेट होना चाहिए डेवलप करते समय. उसके बाद डोसेज फॉर्म है, डिलीवरी सिस्टम है, मतलब इमीडिएट रिलीज है, मॉडिफाइड रिलीज है, सस्टेन्ड रिलीज है, डोसेज स्ट्रेंथ है, उसके बाद कंटेनर क्लोजर सिस्टम कौन सी यूज करनी है. सो ऑल दैट इज पार्ट ऑफ अ QTPP. Next is critical quality attributes. A CQA is a physical, chemical, biological or microbiological property or characteristic that should be within an appropriate limit, range or distribution to ensure the desired product quality. CQAs are generally associated with the drug substance, excipients, intermediates and drug product. So here CQA, for example, CQAs of solid dosage forms are typically those aspects affecting the product quality, product purity, strength, drug release and stability. So these CQAs typically affect these parameters. For drug substances, raw materials and intermediates, the CQAs can additionally include those properties, example particle size distribution, bulk density, that affect drug product CQAs. So these are the CQAs of API or intermediates which can influence the drug product quality. मतलब जैसे हमने पहले डिस्कस किया कि पार्टिकल साइज हो सकता है, टैप डेंसिटी है, बल्क डेंसिटी है, उसके बाद है हार्डनेस है, फ्राइबिलिटी है. So this will be CQAs of drug product, CQAs of drug product or drug substance. Solubility है. Then critical process parameter. A process parameter whose variability has an impact on the critical quality attribute and therefore should be monitored or controlled to ensure the process produces the desired quality. So can anyone tell me the example of critical process parameters for any dosage form?
Dissolution.
Dissolution is a critical process parameter. Dissolution is not a critical process parameter. Dissolution is a critical quality attribute. Process मतलब मैन्युफैक्चरिंग प्रोसेस के दौरान ये पॉइंट क्रिटिकल है और वो हमको मॉनिटर करना है.
पार्टिकल साइज.
हाँ, पार्टिकल साइज है. उसके बाद मिक्सिंग है, मिक्सिंग टाइम है. उसके बाद हार्डनेस है. हार्डनेस के जरिए डिसोल्यूशन पे इम्पैक्ट हो सकता है या फ्राइबिलिटी पे इम्पैक्ट हो सकता है. सो दीज आर द मशीन पैरामीटर्स. मतलब एक तरह से वो प्रोसेस पैरामीटर्स है. उसके बाद है पीएच. पीएच जो है इन बिटवीन मैन्युफैक्चरिंग प्रोसेस, हम अगर पीएच प्रॉपर मेंटेन नहीं कर सकते हैं, नहीं मतलब हमने नहीं किया, तो फिर आगे वो जल्दी मतलब स्टेबिलिटी उसकी ज्यादा नहीं होगी उस प्रोडक्ट की. Loss on drying is a critical process parameter for the products which tend to degrade because of the presence of higher amount of water. ये possible है. So these are the critical process parameters. मतलब ये process parameters कंट्रोल करना जरूरी है क्योंकि ये इसका इम्पैक्ट ड्रग प्रोडक्ट की क्वालिटी पे है. अभी डोसेज फॉर्म, मैंने जैसे पहले भी बताया कि डोसेज फॉर्म के अकॉर्डिंग ये वेरी होते हैं क्रिटिकल प्रोसेस पैरामीटर्स. अगर च्यूएबल टैबलेट है तो हार्डनेस इज अ क्रिटिकल प्रोसेस पैरामीटर्स. अगर लो डोसेज फॉर्म प्रोडक्ट्स है तो मिक्सिंग इज अ प्रोसेस पैरामीटर, क्रिटिकल प्रोसेस पैरामीटर. उसके बाद स्टराइल प्रोडक्ट्स है तो स्टरलिटी जो आप ये कर रहे हैं, मॉनिटर कर रहे हो, तो वो प्रोसेस पैरामीटर क्रिटिकल हो सकता है ड्रग प्रोडक्ट के परफॉर्मेंस के लिए. माइक्रोबियल ग्रोथ हो सकती है, इसके लिए हमको स्टरलिटी इंश्योर करनी है, तो दैट विल बी द प्रोसेस पैरामीटर. अब स्टरलिटी इंश्योर करनी है तो उसमें प्रोसेस पैरामीटर्स कौन कौन से रहते हैं? टेम्परेचर रहता है, प्रेशर रहता है, हीट रहता है, वो सब पैरामीटर्स है. सो लाइकवाइज दोस प्रोसेस आर क्रिटिकल क्वालिटी एट्रीब्यूट. मतलब पार्टिकल साइज हो गया, पॉलीमॉर्फिज्म हो गया, सोलिबिलिटी हो गया. प्रोसेस मतलब मैन्युफैक्चरिंग प्रोसेस में वो कंट्रोल करना जरूरी है. और क्वालिटी एट्रीब्यूट मतलब स्पेसिफिकेशन में जो टेस्ट है, स्पेसिफिकेशन में कौन से टेस्ट रहते हैं? एपीआई एक्सिपिएंट के, अपीयरेंस रहता है, सोलिबिलिटी रहता है, उसके बाद ऑप्टिकल रोटेशन है, पार्टिकल साइज है, टैप डेंसिटी, बल्क डेंसिटी है, पॉलीमॉर्फिज्म है, माइक्रोबियल है. तो ये हो गए क्रिटिकल क्वालिटी एट्रीब्यूट्स. सो देयर इज अ डिफरेंस. The next is quality. What is the definition of quality? The suitability of either a drug substance or a drug product for its intended use. Then control strategy. Control strategy is designed to ensure that a product of required quality will be produced consistently. The control strategy is designed to ensure that a product of required quality will be produced consistently. So that is called as a control strategy. Then a control strategy can include but is not limited to the following. Control of input materials attributes, example drug substance, excipients, primary packaging materials, based on an understanding of their impact on processability or product quality. So first we need to control the input material attributes. Then we need to control the product specification. Then control of for unit operations that have an impact on downstream processing or product quality. Example, the impact of drying on degradation, particle size distribution of the granulate on dissolution. So we have to control the process. Then in process or real-time release testing. example, measurement and control of CQS during the processing. Then a monitoring program, example, full product testing at regular intervals for verifying multivariate prediction models. So this is the control strategy we need to follow while manufacturing a product. Control strategy, why it is important? Control strategy is important because it helps us to मतलब कंट्रोल स्ट्रेटजी अगर हमने अप्लाई की तो इट इज डेफिनेट दैट वी विल गेट अ गुड क्वालिटी प्रोडक्ट. अगर हम कुछ कंट्रोल स्ट्रेटजी अप्लाई नहीं करेंगे, मतलब हम जो हम मटेरियल्स यूज करने वाले उसका हम क्वालिटी अगर मॉनिटर नहीं करेंगे, जो मैन्युफैक्चरिंग प्रोसेस हम फॉलो कर रहे हैं उसमें अगर बीच बीच में हम टेस्ट नहीं परफॉर्म करेंगे, इन प्रोसेस टेस्ट, तो हमको कौन सा क्वालिटी प्रोडक्ट, मतलब कैसा प्रोडक्ट मैन्युफैक्चर होने वाला वो हम गारंटी नहीं दे सकते. बराबर. अगर कुछ सपोज अभी इन प्रोसेस टेस्ट हम कर रहे हैं उसमें अब हमको अगर कुछ क्वालिटी में प्रॉब्लम अगर वी आइडेंटिफाई, देन वी कैन करेक्ट एट दैट टाइम ओनली. अगर हम डायरेक्टली इन प्रोसेस नहीं करेंगे और डायरेक्टली फिनिश प्रोडक्ट का टेस्टिंग करेंगे और उसमें अगर हमको कुछ प्लस माइनस दिखा तो हम आइडेंटिफाई नहीं कर पाएंगे कि ये स्टेप में हमको प्रॉब्लम हो रहा है, इसके लिए द रिजल्ट्स ऑफ द फाइनल प्रोडक्ट आर deviating, so that we not predict at that moment. So that's why in process testing is important as part of a control strategy. Then control of unit operations, मतलब जो unit operations है जिसका impact है product performance पे, that unit operations also we need to check. Then a monitoring program, full product testing at regular intervals for verifying the there are multiple control strategies which we can implement to get a good quality product. Any doubt in this? Move to the next slide. So here we will finish the quality guidelines of the ICH. We have seen Q1 guideline regarding the stability, Q2 for the validation, Q3 for the impurities, then Q6 for the specification, and Q8 for the pharmaceutical development report. So these are the important guidelines related to the regulatory affairs. Okay. Other than that, if you experience any guideline which I need to explain, then you can tell me. I will explain you that guideline. No problem. But now we will move ahead with the safety guidelines of the ICH. Safety means the preclinical studies which will be performed on the animals to check the safety of the product. These safety studies reports are submitted in the module 4 of the CTD dossier. This data is taken care by the experts which are from the medical background. Okay, they will perform the studies and give us ready-made reports that we need to attach in the dossier. For the new products, this preclinical study is usually performed, but for the generic products, the preclinical studies are not performed. Directly, the literature references of the reference product or innovator products are taken from the scientific journals, and those are enclosed in the module 4 of the dossier. मतलब जेनरिक प्रोडक्ट्स के लिए प्रीक्लिनिकल स्टडीज परफॉर्म नहीं की जाती. So that's why here, as a regulatory person, we don't need to perform any activity. We just need to coordinate with the preclinical experts, and we get the reports, and that we will attached in the dossier. And if any queries arise on that documents by the authority, then we need to ask the justification or the response from the preclinical experts. So that's why I've just enlisted this guideline sequentially. I will not go into deep. ICH has produced a comprehensive set of safety guidelines to uncover potential risks like carcinogenicity, genotoxicity, reproductive toxicity. So these are the toxicity studies which are performed to check whether the material is safe or not. So these are the safety studies. The next is the efficacy studies. Efficacy guidelines are used, मतलब ये guidelines मतलब मॉड्यूल 5 जो रहता है डोजियर का, उसमें क्लिनिकल स्टडीज या बायो इक्विवेलेंट स्टडीज सबमिट होती हैं, बराबर. There is also one expert team of doctors which is performing the bioequivalent studies. Or some of the companies, what do they hire one CRO, that is a clinical research organization, and they give a contract to that company and also the product of test and reference product, and they pay the money to that CRO, and the CRO will perform the bioequivalent study and provide the report to the company, okay, generic company. And in this way, some companies are now performing the BE studies, because maintaining the BE site of its own, it is a very high cost. So that's why most of the companies are doing now taking the help of CROs for this. So here also, as a regulatory person, we do not need to perform any means review or any activity. Just we need to attach the report which is received from the medical team, and we need to submit it in the dossier. And if any queries arise by the authority, then we need to ask the justification from the expert team. So in this way, this section is prepared. The work carried out by the ICH under the efficacy heading is concerned with the design, conduct, safety, and reporting of clinical trials. So these are the efficacy guidelines. In this guideline, we will go through the two guidelines. One is the in this efficacy guideline there is no. These are the efficacy guidelines. In the multidisciplinary guidelines, we will see two guidelines mainly. That is a M4 common technical document, which is related to the format of the dossier. Okay, and M9, that is a biopharmaceutical classification system based biowaivers. So these two guidelines we will see in the multidisciplinary section. Multidisciplinary guidelines are the guidelines mainly on the different topics. मतलब सिर्फ ये जैसे क्वालिटी सेफ्टी एफिकेसी में कैसा है कि वो डिवाइड हुआ है बेस्ड ऑन दी क्वालिटी सेफ्टी एंड एफिकेसी टॉपिक, लेकिन मल्टीडिसिप्लिनरी बहुत सारे टॉपिक्स के ऊपर गाइडलाइंस है और वो फिर एक ही सेक्शन के अंदर वो प्लेस किया है, मतलब मल्टीडिसिप्लिनरी, मतलब डिफरेंट टॉपिक्स के ऊपर वो गाइडलाइंस आईसीएच ने डेवलप किए हैं. सो इन दैट these are the topics which do not fit uniquely into the quality, safety, and efficacy categories. It includes the ICH medical terminology, that is MED-DRA, the common technical document, and the development of electronic standards for the transfer of regulatory information, ESTRI. So these are the different topics under the multidisciplinary. So here we will see M4 and M9. So what is mean by QTPP?
Quality targeted product profile.
Yes. Quality target product profile, and it is calculated based on the analysis of reference product, and then the standards are set, and according to the standards, then generic product development is initiated. Yes. Then second is, what is CQA?
Critical quality attributes.
Yes, right. Critical quality attributes. And then next is, what is CPP?
Critical process parameter.
Yes. So, and next is, what is the definition of quality?
Is either drug substance or drug product for its intended use.
Yes, suitability of the drug substance or drug product for its intended use. Yes. Then what is a control strategy? What do you mean by control strategy?
It is nothing but ensuring that a product of required quality is produced consistently.
Yes, it is ensured that yes, quality product is developed consistently with the help of some control measures. Yes, right. So next guideline we will see here, M9 guideline. First we will see, that is a biopharmaceutics classification system based biowaivers. Can anyone tell me what is mean by the BCS classification?
Yes, sir.
Yes.
It is a system in which drugs are classified on the basis of solubility and permeability.
Yes, right. Drugs are classified based on the solubility and permeability, right? So here we will see the introduction. Two drug products containing the same drug substance are considered bioequivalent if their bioavailabilities, that is the rate and extent of drug absorption after administration in the same dose, lie within acceptable predefined limits. So bioequivalent, दो प्रोडक्ट्स बायोइक्विवेलेंट है, हम कभी मतलब बोल सकते हैं जब भी वो दो प्रोडक्ट्स के बायो अवेलेबिलिटी एक साथ मैच होते हैं. बायो अवेलेबिलिटी मतलब रेट एंड एक्सटेंट ऑफ अवेलेबिलिटी ऑफ द एपीआई एट द साइट ऑफ एक्शन. A biowaiver means that in vivo bioavailability and or bioequivalent studies may be waived, not considered necessary for product approval. Biowaiver. अभी biowaiver means suppose I will go through the first classification, BCS classification, then I will explain you what is meant by biowaiver. The BCS classification based biowaiver approach is intended to reduce the need for in vivo bioequivalence studies. That is, it can provide a substitute for in vivo bioequivalence. In vivo bioequivalence studies may be exempted if an assumption of equivalence in vivo performance can be justified by satisfactory in vitro data. The BCS is a scientific approach based on the aqueous solubility and intestinal permeability characteristics of the drug substances. The BCS categorizes drug substances into one of four BCS classes. So we will see the classification. There are four classes. Class 1 is high solubility, high permeability. Class 2 is low solubility and high permeability. Class 3 is high solubility, low permeability. And class 4 is low solubility and low permeability. So in this way, the drug substances are classified into 4 classes. The BCS-based bioweaver is only applicable to immediate release, solid orally administered dosage forms or suspensions designed to deliver drug to the systemic circulation. So the drug products which deliver the drug to the systemic circulation and which are immediate release in nature and which are solid orally administered dosage forms or suspensions, those can be applied to the BCS-based bioweaver. Drug products having a narrow therapeutic index are excluded from consideration for a BCS-based bioweaver in this guideline. So narrow therapeutic index, can anyone tell me what is meant by narrow therapeutic index? Huh?
It means in small quantity, but there is an means if drug in smaller concentration can also lead to serious therapeutic conditions, adverse effects.
Yes, right. So there is a less difference between the therapeutic dose and lethal dose. That is called as a narrow therapeutic index. मतलब कुछ थोड़ा भी अगर concentration increase हो जाता है, dose increase हो जाए, तो वो मतलब patient की मतलब that can be lethal to the patient. Lethal मतलब patient की जान भी जा सकती है. So or more adverse effects will occur that patient. So there is a less difference between the therapeutic dose and lethal dose. That is called as a narrow therapeutic index dose. So BCS-based bioweavers are applicable to the drug products where the drug substances exhibit high solubility and either high permeability or low permeability. That is a BCS class 3. मतलब BCS class 1 और BCS class 3 के लिए bioweaver approach applicable है. अभी I will tell you what is mean by bioweaver. Bioweaver मतलब अगर suppose जो एपीआई हम यूज कर रहे हैं फॉर्मुलेशन में वो अगर बीसीएस क्लास 1 का है या 3 का है तो हमको इक्विवेलेंस प्रूव करने के लिए मतलब रेफरेंस प्रोडक्ट और जेनरिक प्रोडक्ट में इक्विवेलेंस मतलब कि बाबा दोनों का रेट एंड एक्सटेंट ऑफ अवेलेबिलिटी एट द साइट ऑफ एक्शन इज सिमिलर. ये प्रूव करने के लिए हमको बायो इक्विवेलेंस स्टडी परफॉर्म करने की जरूरत नहीं है. हमको सिर्फ इन विट्रो स्टडी परफॉर्म करने की जरूरत है. इन विट्रो स्टडी मतलब इन विट्रो डिसोल्यूशन स्टडी. सो इससे क्या होगा कि हमारा बायो इक्विवेलेंस का पैसा बच जाएगा और हम डोजियर जल्दी फाइल कर सकते हैं. सो दैट इज द यूज ऑफ bioweaver. That is the use of BCS classification. So the APIs which are falling under class 1 and class 3, the bioweaver approach can be applied to those. Okay. So for those APIs, bioequivalence study is not required to be performed. Only in vitro dissolution study is sufficient. And for other two classes, that is a class 2 and class 4, if the API is falling under these classes, then we have to perform the bioequivalence study. And that study report we need to submit in the module 5. A biowaiver is applicable when the drug substance in test and reference product are identical. So this is obvious. A biowaiver may also be applicable if test and reference products contain different salts, provided that both belong to BCS class 1, high solubility and high permeability. A biowaiver is not applicable when the test product contains a different ester, ether, isomer, mixture of isomers, complex or derivatives of drug substance from that of the reference product, since these differences may lead to different viability. Means whenever we are applying a biowaiver, we need to check whether the API form is matching or not. मतलब जो salt form है तो रेफरेंस प्रोडक्ट में जो salt form है वो भी जनरल प्रोडक्ट में होना चाहिए. मतलब कुछ भी डिफरेंस ज्यादा नहीं हो. अगर डिफरेंस है भी तो वो डिफरेंस ज्यादा नहीं होना चाहिए, so that it will affect the bioequivalence or it will affect the bioavailability. So that needs to be taken in care. जो ईस्टर है, इथर है, आइसोमर है, वो नहीं है, डिफरेंट नहीं होना चाहिए, बिकॉज इट विल अफेक्ट द बायो अवेलेबिलिटी. अभी ये सोल्यूबिलिटी और परमेबिलिटी के बेसिस पे एपीआईज को डिवाइड किया, मतलब क्लासिफाई किया. सो हाउ दिस हाई सोल्यूबिलिटी, हाई परमेबिलिटी इज डिसाइडेड. So a drug substance is classified as highly soluble if the highest single therapeutic dose is completely soluble in 250 ml or less of aqueous media over the pH range of 1.2 to 6.8 at 37 degrees Celsius. At least 3 pHs within the range, including buffers at pH 1.2, 4.5 and 6.8 should be evaluated. So here we can see हाईएस्ट सिंगल थेरेपेटिक डोज, मतलब एक ही टाइम पे हम हाईएस्ट सिंगल थेरेपेटिक डोज कितना लेते हैं, वो एपीआई का 100 mg रहेगा, 200 mg का रहेगा, अगर डायबिटिक प्रोडक्ट है तो 500 mg या उससे भी ज्यादा रहेगा. सो इन दिस वे, depending on the API, what is the highest therapeutic dose we are taking, okay, that should be completely dissolved within 250 ml of aqueous media over the pH of 1.2 to 6.8 at 37 degrees. अभी 1.2 to 6.8 क्यों लिया है, क्योंकि हमारा वो बायोलॉजिकल जो फ्लूइड है, मतलब 1.2 is very much similar to the stomach acid, acidic fluid है. उसके बाद है 4.5 which is somewhat to the intestine, then 6.8 is the large intestine. So in this way, these pHs are dependent on our biological fluid pH. At least 3 pH within this range, including buffers at pH 1.2, 4.5 and 6.8 should be evaluated. The next is permeability. The assessment of permeability should preferentially be based on the extent of absorption derived from human pharmacokinetic studies. So permeability कैसे determine होता है, तो जो cell lines रहती है, मतलब एक्सटर्नल जो सेल लाइंस होती है, जिसके बेसिस पे ये परमेबिलिटी डिसाइड की जाती है कि हाईली परमेबल है कि नहीं, यहाँ कितना परमेबल है. Example, absolute bioavailability or mass balance. High permeability can be concluded when the absolute bioavailability is more than or equal to 85%. High permeability can be concluded when the absolute bioavailability is more than or equal to 85%. So this is a permeability. In this way, the permeability is determined with the help of the external cell lines. Any doubt in this section? Next, for BCS class 1 drugs, qualitative and quantitative differences in excipients are permitted, except for excipients that may affect the absorption, which should be qualitatively the same and quantitatively similar, that is within plus or minus 10% of the amount of excipient in the reference product. So प्लस और माइनस 10% तक डिफरेंसेस एक्सेप्टेबल है अगर बीसीएस क्लास 1 ड्रग है तो. Additionally, the cumulative difference for excipients that may affect absorption should be within plus or minus 10%. मतलब हर एक एक्सिपिएंट का प्लस और माइनस डिफरेंस नहीं, that is not acceptable. क्यूमुलेटिव मतलब अगर सपोज हम 5 एक्सिपिएंट्स यूज कर रहे हैं तो 5 में से सभी एक्सिपिएंट्स डिफरेंट होंगे, तो दैट विल आल्सो नॉट सर्व पर्पस. उसमें से अगर 2 एक्सिपिएंट्स डिफर हो रहे हैं क्वालिटेटिवली और क्वांटिटेटिवली, मतलब उसका सॉल्ट फॉर्म है या क्वांटिटी कुछ तो डिफरेंट है, लेकिन वो दोनों के डिफरेंसेस मिला के 10% के ऊपर नहीं जाना चाहिए वो डिफरेंस, देन ओनली वी कैन अप्लाई द बायोवेवर फॉर द बीसीएस क्लास 1 ड्रग्स. सो ये जो क्यूमुलेटिव डिफरेंस है वो 10% से ज्यादा नहीं होना चाहिए. For BCS class 3 drugs, all of the excipients should be qualitatively the same and quantitatively similar, except for film coating or capsule shell excipients. Excipients that may affect absorption should be qualitatively the same and quantitatively similar, that is within plus or minus 10% of the amount of excipient in the reference product, and the cumulative difference for these excipients should be within plus or minus 10%. So in this way, for BCS class 3 drugs also, the cumulative difference should not be more than 10%. Then drug products with buccal or sublingual absorption are not eligible for a BCS based biowear application. So this, if the dosage form is a buccal or sublingual, then those are not applicable for the BCS based biowear application. Furthermore, BCS based biowear approach is applicable only when the mode of administration includes water. If administration without water is also intended, example orodispersible products, a bioequivalence study in which the product is dosed without water should be conducted. So here, these are the criteria which we need to follow while applying the BioWaiver criteria, BioWaiver approach. बकल और सबलिंगुअल के लिए एप्लीकेबल नहीं है क्योंकि वो डायरेक्टली, मतलब वो they do not dissolve in the stomach or intestine. They directly absorbed from the buccal or sublingual way. Right, so that's why this dissolution type of pattern will not be followed there. So bioequivalence study करना ही important है उसमें. उसके बाद जिसमें water use होता है, those only are applicable for the BioWaiver. If administration without water is also intended, a bioequivalence study in which the product is dosed without water should be conducted. So in this way, these criteria should be fulfilled, then only we can apply the BioWaiver approach. So here are two types of bioavailabilities. One is absolute, second is relative. Absolute means when the systemic availability of an orally administered drug is calculated in comparison to its intravenous administration, it is called as absolute bioavailability. मतलब सपोज ओरल रूट ऑफ एडमिनिस्ट्रेशन का एक ड्रग है और उसका बायोअवेलेबिलिटी इंट्रावेनस रूट के डोसेज फॉर्म के साथ कंपेयर किया, then that is called as an absolute bioavailability, because we are comparing to a product which is having an intravenous administration and which is 100% bioavailability रहती है, इंट्रावेनस के जो डोसेज फॉर्म्स रहते हैं उनकी 100% बायोअवेलेबिलिटी रहती है, उसके इसके लिए उसको एब्सोल्यूट बायोअवेलेबिलिटी मतलब जब भी कंपैरिजन करते हैं तभी. And relative bioavailability means when the systemic availability of a drug after administration is compared with that of a standard of the same drug, it is known as relative bioavailability. Means oral route of administration का बायोअवेलेबिलिटी दूसरा जो रेफरेंस प्रोडक्ट है वो भी ओरल रूट का ही है, तो उसके साथ अगर कंपेयर किया तो दैट इज कॉल्ड एज अ रिलेटिव बायोअवेलेबिलिटी. सो दीज आर द टू टाइप्स ऑफ बायोअवेलेबिलिटीज. क्वेश्चंस मतलब ये जो मतलब नॉर्मल क्वेश्चंस है कि वो टेल मी द डेफिनेशन ऑफ बायोअवेलेबिलिटी, टेल मी द टाइप्स ऑफ बायोअवेलेबिलिटीज, देन द डेफिनेशन ऑफ बायोइक्विवेलेंस, what is the BCS classification, tell me the classification of 4 classes, on which basis they are classified. So these are the normal questions which will be asked on this guideline. What is the meaning of BioWaiver? BioWaiver most commonly पूछा जाता है क्वेश्चन, क्योंकि ये मतलब इंडस्ट्री भी चाहती है कि ज्यादा से ज्यादा प्रोडक्ट्स BioWaiver अप्रोच को में हम अप्लाई कर सके, क्योंकि दैट इज अ वेरी कॉस्ट सेविंग अप्रोच. बाय इक्विवेलेंस हम स्किप कर सकते हैं. So that is very helpful to the authority as well, helpful to the industry and as well as to the authority as well, because they are also getting an means उनको भी जल्दी डोजियर रिव्यू करके वो प्रोडक्ट पब्लिकली अवेलेबल कर सकते हैं. टाइम एंड मनी दोनों भी सेव होता है इसमें. नेक्स्ट इज इन विट्रो डिसोल्यूशन. अभी हमको अगर बायोवेवर फॉलो करना है तो हमको इन विट्रो डिसोल्यूशन स्टडी परफॉर्म करनी जरूरी है. उसके बेसिस पे ही हम प्रूव कर सकते हैं कि बाबा हम टेस्ट प्रोडक्ट जो है वो रेफरेंस प्रोडक्ट के अकॉर्डिंग मतलब डिसोल्यूशन प्रोफाइल दोनों का मैचिंग है, कंपेरेबल है. When applying the BCS based BioWaiver approach, comparative in vitro dissolution test should be conducted using one batch representative of the proposed commercial manufacturing process for the test product relative to the reference product. At least 12 units of reference and test product should be used for each dissolution profile determination. 12 units means 12 tablets should be used of each, that is a reference and test. मतलब total 24 tablets use होंगी in vitro dissolution के लिए. 3 buffers 1.2 pH, 4.5 and 6.8. Sorry. Then to qualify for a BCS based BioWaiver, for BCS class 1 drug substances, both the test product and the reference product should display either very rapid, that is more than or equal to 85% of the mean percent dissolved in less than or equal to 15 minutes, in vitro dissolution characteristics, or rapid, that is more than or equal to 85% for the mean percent dissolved in less than or equal to 30 minutes, and similar in vitro dissolution characteristics, that is based on F2 comparison under all of the defined conditions. So this is the means अगर क्वालिफाई करना है BCS बेस्ड बायोवेयर के लिए तो इतना समय में वो डिसॉल्व होना चाहिए और इतने परसेंटेज में. सिर्फ BCS क्लास 1 है और क्लास 3 है, इसके लिए बायोवेयर एप्लीकेबल नहीं हो सकता. उसके साथ इन विट्रो डिसोल्यूशन में ये मोर देन 85% विद इन 15 मिनट्स डिसॉल्व होना चाहिए और मोर देन 85% विद इन 30 मिनट्स डिसॉल्व होना चाहिए, तो ही हम उसको बायोवेयर अप्लाई कर सकते हैं. सिर्फ लिटरेचर रेफरेंसेस के जरिए हमने आइडेंटिफाई किया कि बाबा ये BCS क्लास 1 का API है तो हम डायरेक्टली इसको बायोवेयर अप्लाई कर सकते हैं. लेकिन बायोवेयर अप्लाई करना है मतलब उसका इन विट्रो डिसोल्यूशन स्टडी हमको परफॉर्म करना है, और इन विट्रो डिसोल्यूशन स्टडी करने के बाद हमको रिजल्ट्स ऐसे मिलने चाहिए कि बाबा वो more than 85% dissolve होना चाहिए within 15 minutes or more than 85% dissolve होना चाहिए within 30 minutes. Then only we can consider that it is applicable for the BioWare approach. Otherwise, it is not. There are two factors which will be calculated at the end of this in vitro dissolution study. One is the F1 factor. It is also called as a dissimilarity factor. The result of this factor should come means standard जो है less than or equal to 15 आना चाहिए. And similarity factor, that is F2, it should come as more than or equal to 50. Similarity factor means the dissolution profiles are comparable. मतलब 50 के ऊपर अगर रिजल्ट आता है तो दोनों के डिसोल्यूशन मतलब टेस्ट और रेफरेंस प्रोडक्ट के डिसोल्यूशन प्रोफाइल्स कंपेरेबल है. इट इज अ इंडिकेशन ऑफ दैट. मतलब 50 के ऊपर अगर वैल्यू ऑब्जर्व होता है तो हम बोल सकते हैं कि बाबा दोनों के डिसोल्यूशन प्रोफाइल्स कंपेरेबल है. और सिमिलरली अगर 50 के नीचे आता है तो डिसोल्यूशन प्रोफाइल्स कंपेरेबल नहीं है. F1 के लिए भी सेम है. 15 के ऊपर अगर आता है तो डिसोल्यूशन प्रोफाइल्स कंपेरेबल नहीं है. Here are some questions. So definition of BioWare. What is the definition? Yes, anyone? BioWare का definition. Yes, anyone?
Yes, sir.
Yes.
It means that in vivo viability or bioequivalence studies may be considered for the product approval. I mean,
No. What you said? Can you repeat?
it is in vivo viability and or bioequivalence studies may be considered for the product approval.
No. BioWare मतलब it is not considered necessary to perform the bioequivalence study for the approval of the product. मतलब अप्रूवल डोजियर अप्रूवल के लिए या प्रोडक्ट अप्रूवल के लिए इन विवो बायोइक्विवेलेंस स्टडी जरूरी नहीं है. क्यों जरूरी नहीं है? क्योंकि BioWare अप्रोच इज एप्लीकेबल फॉर दैट ड्रग प्रोडक्ट. I will show you once again the definition so you will able to understand this. Yes, here is the definition. Can you able to see this? A BioWare means that in vivo bioavailability and or bioequivalence studies may be waived. Waived का meaning है not considered necessary for product approval. मतलब in the cases where the in vivo bioequivalence studies are not considered necessary for product approval, that case is considered as a BioWare approach. And what is the BCS classification?
Hello.
Yes.
It is a system in which drugs are differentiated on the basis of solubility and permeability.
Yes. drugs are means they are differentiated based on the solubility and permeability. How solubility study is performed? Here the highest therapeutic dose is dissolved in 250 ml of aqueous media over the pH range of 1.2 to 6.8. So that should be completely soluble. So in this way the solubility is identified. And differences between the absolute and relative bioavailability is, yes, anyone?
Absolute bioavailability means okay. Hello.
Yes.
Absolute bioavailability means comparison in between the oral administered drug and intravenous drug administration.
Yes, right.
In a relative bioavailability, drug is compared is between the standard with the same drug.
Dosage form.
Yes, sir.
Yes. Then what is in vitro dissolution study? Yes, Ratuja. What is in vitro dissolution study?
In vitro dissolution study means when applying the BCS, which is based on biowaiver approach, which is compared to in vitro dissolution test, should be conducted.
In vitro dissolution means we perform the dissolution study of test product versus the reference product, and we compare that the dissolution profiles of both the products are equivalent to each other or not based on the F1 and F2 factor. There is one calculation for this, one formula is there to calculate the F2 factor. I will share one PDF. In that dissolution related data of the means it is explained. So it is a very detailed guideline. So you can read that. You will better understand how this is done and what are the criteria. So in detail may explain kiya hai. So I suggest you to go through that. You will get a good understanding. Yes. Okay. Yes. Any doubts? From the next session, we will start with the CTD dossier structure, and so I expect the maximum attendance. And from the next Saturday session, we will start the mock interviews. So all the syllabus which is completed till now will be considered for this, and all the students can start preparing this for this mock interviews, and try to complete your tasks as early as possible. And for the students of which topics are completed for PPT, those students should complete their PPT presentation, because at the last minute the hurry will occur, and means you will not get the time to get PPT present. So for the candidates whose topics are covered, so please present the PPTs. We have the limited sessions available, and in that also we have to take the mock interviews and PPT also. Till now only 3 students have given the PPT presentation. I think 10 to 12 students topics are completed, and so start giving the PPT presentation from the next Saturday. Also mock interviews are also started, so start preparing for that. So any doubts?
Sir, from coming Saturday onwards, sir, mock interviews going to be
Yes,
conducted?
yes. Coming Saturday.
Okay, sir.
Any other doubt?